Drucker. I'm going to chair this last session, and our next speaker is Professor Jeffrey Flier. Some of you know Jeff from his roles as an endocrinologist doing seminal work on insulin resistance and metabolism, and later on leptin biology. And then he had a little gig as Dean of Harvard Medical School, which he's now stepped down from. I first met Jeff, he doesn't like me to say this, I first met him in 1979 when I was doing an elective at the Beth Israel Hospital. So I've known him a long time and looked up to him and learned a lot from him. And he will take us through an important chapter of the modern day insulin research discovery and what we've learned since then. Thanks, Jeff.
Okay, so thank you all for inviting me to this important meeting of the Toronto Medical Historical Club. I want to thank John Dirks and others for inviting me to give a lecture today on the topic of the pace-setting science of insulin in the decades after its discovery. Now, everyone knows that the discovery of insulin in 1921 in Toronto had an immense effect on modern bioscience, but it also had ongoing effects that continue up until today. And my job is to tell you about some of the high points of those subsequent discoveries. I will do it in 7 brief stories. The first occurred between 1951 and 1955. And this was the discovery of the molecular identity of insulin as a disulfide-linked 2-chain molecule of defined amino acid composition by Sanger. The second occurred in 1960, a key discovery, the measurement of insulin in blood by a new technique named radioimmunoassay by Burson and Yallo. Third, the synthesis of insulin to confirm its proposed structure and permit the testing of novel sequence variations of the insulin molecule by Katsayanis and Zahn. Then number 4, in 1967, the discovery of prohormone processing, how insulin is synthesized initially as a single chain precursor called proinsulin by Don Steiner. Then in 1969, the crystal structure of insulin is solved by Hodgkin, Blundell, and Dodson. In 1977, going on to 1983, the cloning and expression of the insulin gene, and insulin as the first recombinant human therapeutic by many people, but most importantly, Rudder, Goodman, and Gilbert. And then to end up, starting in 1971 or so, and really going on until today, studies of the insulin receptor as a paradigm for plasma membrane receptors and membrane signaling, Roth, Quattrocassis, Ulrich, Kahn, and others. These 7 discoveries, which really, as you can see here, began around 30 years after the initial discovery of insulin, went on for 30 plus years, and they placed insulin at the center of modern bioscience during this period, going way beyond the specifics of insulin and diabetes. So let's start with the elucidation of the amino acid sequence and structure of insulin. The leading scientist here was Fred Sanger, and next to him, the iconic depiction of the linear amino acid sequence of insulin, a monomer of 51 amino acids, an A chain with 20 amino acids, a B chain with 31, and 3 disulfide bonds. Now let's remember that at the time of its discovery and for years afterwards, what insulin actually was unknown. It was known it was something you could reproducibly extract from the pancreas, and when you injected it into patients with diabetes, their blood sugars came down. But it wasn't clear what it was. This was in the early period of the field of endocrinology. Evidence began to accumulate that it was likely to be a protein. This was from some chemical attributes of the activity and by the fact that a protease treatment would make the activity go away. But Sanger, a young biochemist, thought that he wanted to determine exactly what the insulin structure was. It was an extremely important apparent protein that did not have a known structure. And secondly, because it was being used therapeutically, it was commercially available and he didn't have to purify it all the time by himself. This was not an easy job, however. He had to develop multiple new technologies to enable insulin to be sequenced. Among those were N-terminal sequencing with a reagent called DNFB, which is now termed Sanger's reagent. This allows N-terminal sequencing of amino acids. He would then, by various approaches, hydrolyze the protein, fractionate it by novel approaches, and get the N-terminal sequences, and then using fingerprints for the individual peptides technique that he developed, he was able to deduce the sequence of the A and the B chains and ultimately the disulfide bonds. Insulin was the first protein to be sequenced, and this was published definitively in 1952 and established for the first time that proteins had a defined structure. Now, we may think today everybody always knew that proteins had a defined structure, but no, they didn't. And people thought maybe they're these amorphous associations of amino acids, and how they had their definitive uniqueness was not clear. For this work, and most deservedly, Sanger received the Nobel Prize in Chemistry in 1958. Amazingly, and showing how brilliant he was, he won a second chemistry Nobel Prize in 1980 for his discovery of dideoxy sequencing of DNA, and he shared it with Gilbert and Berg. This method was very rapidly applied to sequencing the insulin gene. So you can see that there are multiple ways that insulin comes into this story. Sanger lived to the age of 95. When he died in 2013, his obituary in the Times of London quoted him earlier on as describing himself as just a chap who messed about in a lab. Quite an understatement, I would say. Now let's move on to the measurement of insulin in blood. This is based on the work of Solomon Bersin and Rosalind Yalow, both of whom, as I'll mention, I knew personally. And this is their classic, somewhat understated paper from 1960 in the Journal of Clinical Investigation, Immunoassay of Endogenous Plasma Insulin in Man. I will remind you that following its discovery in 1922, there was no way to measure insulin in the blood or in any other compartment. And many unsuccessful efforts were undertaken, mainly, if not exclusively, with bioassays, but these, not surprisingly, lacked specificity and sensitivity and didn't really reveal anything important about the level of insulin in the blood. Who were these two people, Bersin and Yalow? And I'll just spend a minute telling you a bit. So Saul Bersin grew up in New York City, went to the City College of New York, where I happened to go as well. And while he was there, he was a good student, but he also became a great violinist and a chess master. When he applied to medical school, he applied to many, many medical schools and didn't get in. So he took a couple of years, he did some work in anatomy, he helped teach anatomy at NYU, and then he reapplied and finally got into NYU Medical School. He then went on to train in internal medicine. He had no real formal research training. He ultimately went to the Bronx Veterans Hospital in New York, where the field of radioisotopes was just becoming a significant field, and he decided he was going to do some things with that. There he met Rosalind Yalow, also grew up in New York City. She was a brilliant young lady who wanted to be a physicist. She was discouraged by many people who said, oh, women don't become physicists and they can't get jobs. But she persisted. She ultimately got a job and a PhD as a physicist and took a position at the Bronx VA to help run the radioisotope laboratory, where they met each other, and the rest is history. So what, in fact, did they do? Well, they came up with the idea that they wanted to use the labeling of insulin with radioactive iodine to test the hypothesis that was prevalent at the time. This was the theory of Mirsky, that adult diabetes was caused by excessive degradation of insulin by something that he called an insulinase. This turned out not to be true, but it was a prominent idea, and Burson and Yalow decided to test it. They tested it by making iodinated insulin. They got approval to inject it either into non-diabetic or diabetic individuals, and they would look at the disappearance of the labeled radioactivity. And if there was a degradation process in the insulin-treated diabetics, you would think that the insulin would disappear more rapidly. And they found the opposite. Reproducibly, they found that insulin hung around longer in the blood of the insulin-treated patients. So A, they made that observation. B, they came up with the hypothesis that this could be because they had developed antibodies in their blood to insulin. Most people thought that couldn't happen. They thought it could happen. And they proved that, in fact, there were immunoglobulins in their serum that bound to insulin. They bound with high affinity. And by studying that, they came to a third hypothesis that they could use these antibodies and labeled insulin and unlabeled insulin to measure insulin. And they did that. And this paper shows all of those things in one important paper, including the fact that most people with type 2 diabetes had high levels of circulating insulin. So that was important. And, they had a lot of resistance to publishing the paper, but they did in fact publish it. They called the technique radioimmunoassay. They immediately realized they could apply the same technology to measure anything for which there would be an antibody against the molecule. And it was rapidly used by them and then by others to measure hundreds of molecules that couldn't previously be measured. Tragically, at the age of, I believe, 55, Saul Burson died suddenly. He died while I was a fourth-year medical student at Mount Sinai, where he was the chair of medicine. I was at his funeral, and because he had died, he couldn't win the Nobel Prize, but in 1977, Yalow won the Nobel Prize for this work. I will just note, as I said, Burson was the inaugural chair of medicine at Mount Sinai, where I was in the first entering class in 1968. I visited his lab many times, and just before his death, I went to see him to get his advice about where I might do research training at NIH. And he stopped me immediately and he said, "Jeff, there's only one lab to go to, Jesse Roth. He was my first student and he's studying receptors for insulin, and that's where you should go." And that's what I did. And when he died and we graduated, we dedicated our school yearbook to him in 1972. And I'll read the quote from him that appeared: "In compensation for the almost daily frustrations and frequent failures experienced by the scientific investigator, there must come certain satisfactions. The most rewarding of these is inherent in the compulsive pleasure of seeking and searching, which occasionally culminates in the thrill of perceiving a little corner of the universe into which no one has previously been privileged to glimpse." Okay, let's move on to the synthesis of bioactive insulin, carried out by Panagiotis Katsagiannis in the US, first in Pittsburgh and then in New York, and Helmut Zahn in Germany. Though Sanger identified the protein structure of insulin, as I showed you, there was no way to confirm and prove that this was a fully active molecule. How would you do it? There was no technique available. And that's why Katsagiannis and Zahn, and then a group in China, set out to synthesize the Sanger molecule by then developing techniques of synthetic chemistry. Vincent Duvigneau had won the 1955 Nobel Chemistry Prize for using this synthetic chemistry to synthesize the small peptide hormones, oxytocin and vasopressin, and show that they were active after they were chemically synthesized. The full synthesis of insulin was reported in 63 and 64 in several papers, and then they went on to prove that this molecule that they synthesized had full biological activity in lowering blood sugar in mice. So they did what they set out to do. Many people thought they would win the Nobel Prize for this, but that did not happen. This validated the Sanger insulin sequence and structure and showed that no further modifications were required for activity. They also speculated at the time that chemical synthesis might supplant the use of insulin extracted from animals, because no one knew whether there might be a supply chain problem with that or inadequate production of extracted insulin. And they also began to make, both of their labs, modifications of the insulin sequence by putting in pieces of insulin with different amino acids to begin to look at structure and function. Katsayanis was the inaugural biochemistry chair at Mount Sinai. So the first day of school, I got introduced to both Burson and to Katsayanis. I spent time in his lab as well as a student performing mouse convolution assays to measure the bioactivity of insulin analogs that they were making. Well, how do these insulin chains get made in the body? There are two chains. They're joined by disulfides. Most people thought they were individually synthesized in the cell and then joined together. Don Steiner, who had an MD degree and some minor training in biochemistry at the University of Chicago, decided he was going to take up that issue. And this is his absolutely classic paper, Insulin Biosynthesis, Proinsulin, Insulin, C-peptide, the biosynthesis and a probable precursor of insulin by human islet adenoma in the PNAS in 1967. And I've read this paper recently, and it's really one of the most elegant papers I have ever read. So it turns out, as people started taking samples from blood of patients and doing radioimmunoassay on chromatographically separated serum, they found that some insulin, most of it was of the size that you might have predicted for insulin, but some of it was so-called big insulin, and no one knew what big insulin represented. And he hypothesized that maybe it was a large precursor. And the way he did this was he learned how to do pulse chase labeling with labeled amino acids in vitro, but he needed to have a proper specimen. And he waited around until someone told him there was a patient being operated on for an insulin-producing tumor of the pancreas, so-called insulinoma, and it was going to be removed. So he was able to get some slices from that insulinoma. He took them to his lab. He did this precursor labeling. He isolated the material. He ran it out on various kinds of gels, and he showed that 90% was the small normal insulin, but 10% was the big insulin. And then he showed that by very light treatment with the protease, he could make the big insulin become the small insulin. Rather remarkable. This established the field of protein precursor processing that has then been applied by him and others to innumerable other proteins. And then C-peptide, the part that's cleaved away that isn't part of insulin per se, has served as a valuable tool for assessing insulin synthesis in various circumstances and worked together with Arthur Rubinstein. Okay, how about the three-dimensional crystal structure of insulin? Most important scientist there was Dorothy Hodgkin. You could see the monomer on the right and the hexamer of insulin as it exists in crystal form in the beta cell. Hodgkin advanced X-ray crystallography, initially working with Bernal, and she solved the structures of pepsin, penicillin, and vitamin B12. These were published by 1955, and for that work, she received a chemistry Nobel Prize in 1964. But it turns out her efforts first began with insulin for some of the same reasons that Sanger studied insulin. It was important, and she could get her hands on it. But the problem was the techniques of crystallography then could not work well enough with a molecule of insulin size and complexity. Eventually, after cutting her teeth and getting the Nobel Prize with these other molecules, she went back to insulin, and they published the insulin structure from 67 to 71, a major achievement. Okay, coming to the last two stories, the cloning and expression of the insulin gene and insulin as the first recombinant therapeutic. Key scientists here, Wally Gilbert, Bill Rutter, and Howard Goodman. And as most of you know, in the early 1970s, recombinant DNA technology was the hottest thing in science. Herb Boyer and Stanley Cohn at Stanford and UCSF had developed patents for recombinant DNA cloning. And many saw that the first application of this, of real note, would be to insulin, to make recombinant insulin. In 1973 to 83, there was a serious race between various laboratories to clone and express the insulin gene. The Rutter-Goodman group at UCSF and the Gilbert group at Harvard, which was set back a little bit because the City Council of Cambridge, Mass, where his lab was at Harvard, had a couple of years of not allowing recombinant DNA research to take place. Eventually, the patent from UCSF and Stanford was licensed to Genentech, which then licensed the product to Lilly, and the rest is history. The Nobel Prize in Chemistry in 1980 was shared by Berg for recombinant DNA and by Gilbert and Sanger for DNA sequencing. Gilbert, while working on sequencing and on cloning and expressing, his main thing that he was trying to express was insulin. In 1982, the FDA approved, and in 1983, it hit the market with recombinant human insulin. I'll never forget the day I read this in the New York Times on the front page. This was the first of what is now more than 170 recombinant human therapeutics. And then to end up with the question of how insulin initiates its specific actions on cells. And the picture here is of my research mentor, Jesse Roth, and the famous paper published in 1971 that was handed to me and everyone else who joined the lab. I joined the lab in July of 1974. I was given this paper and told, read it carefully. The paper was Insulin Receptors in the Liver, Specific Binding of 1,2,5-I Insulin to the Plasma Membrane and its Relation to Insulin Bioactivity. Frechet, who was visiting from France, Roth, and Neville, who was a protein chemist in the Institute who helped them with the work. Through 1960s, whether insulin had its primary site of action inside cells or at the membrane was essentially unknown. It was debated, but there was no data to really definitively answer the question. Sutherland and his group discovered cyclic AMP as what they called the second messenger for the hormone neurotransmitter epinephrine, for which he won the Nobel Prize in 1971. But what the first messenger was, that is, how epinephrine or other molecules like insulin, how they actually got the cell to do something wasn't clear. Many people thought that they got into the cell to change some process or other. However, Roth was really interested in this, and as a young scientist at NIH, he showed that insulin and TSH, another peptide hormone that was being studied, that their action on cells was stopped very rapidly when they added antibodies either to TSH or to insulin. So that suggested to them in a paper that they wrote, very nice paper, that, hey, means that the hormone probably starts its action by doing something at the cell membrane. And that's why if you add something that can't get into the cell, you might reverse the action. And then Quattrocassis, amazingly, in the same institute, on the same floor, at the same time, approximately, had another approach. So he had, among other things, while he was a fellow, he had discovered the technique or co-discovered the technique of affinity chromatography, where you take molecules and you attach them covalently to large Sephiroth beads. And he came up with the idea, well, look, if I can attach insulin to Sephiroth beads, and I can do it in a way where the insulin retains its activity, it stays on the beads. We know the beads can't get into the cells. And if that can stimulate insulin action, that'll probably mean that insulin acts on the cell membrane. And in fact, that's exactly what he did. and he had a paper on that in 1969. And when I came to NIH in 74, I had one freezer in the lab that I was that had stuff from Jesse Roth from when he was doing those early experiments. And then there was a freezer in the room down the hall that had Quattrocassis' name on it. Kind of interesting. Following that, Roth and Quattrocassis independently pursued characterization of labeled insulin interaction with membranes and cells, including measuring the affinity of binding, the specificity, the reversibility, and began to study the variation with physiology and disease. In effect, they became somewhat competitors. Roth at NIH and what became the diabetes branch, and Quattrocassis at Hopkins. And I was at the diabetes branch between 74 and 78. Another discovery to mention in this cavalcade was that when I got to NIH, I found that they had some patients that had extraordinarily severe resistance to insulin. And I had the idea that maybe some of these patients had developed an autoimmune response where the antibody was against the receptor. And that turned out to be true. Those antibodies helped prove that, in fact, these receptors that were being measured were biologically critical. And it also became antibodies that were a tool for future discovery. Other very important things that happened in the field where the insulin receptor was shown to be a tyrosine kinase in 1983 by Ron Kahn and his group, and cloned the receptor in 1985, Axel Ulrich in Germany. And then there were a series of papers with genetic loss of function of the insulin receptor in either a pediatric syndrome called leprechaunism or an adult syndrome called the type A syndrome. And was shown that these loss of function mutations of the insulin receptor caused severe insulin resistance. So let me just summarize what I have tried to tell you about what happened over the past 100 years. We started out with insulin discovered in 1921 and began to be administered to patients shortly thereafter. There's a vial of what was called insulin, although no one knew exactly what was in it, and you'd never be able to inject something like that into a human being today. Then 3 years later, we have the first sequence of the amino acids of insulin, proving that this is what nature had designed as a molecule to control blood sugar and other processes. Then 14 years after that, the crystal structure of insulin, looking at the three-dimensional capacity of this molecule in space. Then over many decades, studies of how insulin worked, binding to insulin receptors, stimulating all kinds of intracellular events that are still being studied today. We have the cloning of the insulin gene and its presence as the first therapeutic recombinant protein, a remarkable story linking the discovery in 1921 to the first recombinant human therapeutic. And then now we have tremendously detailed insights into the structure of the insulin receptor. The extracellular domain has been crystallized, and in the lower right of that picture, you see a picture of the insulin molecule sitting in the extracellular domain to which it binds. So we all celebrate the discovery of insulin in Toronto in 1921, and we also celebrate the sequential discoveries by Sanger, Burson and Yalow, Katsayanis and Zahn, Steiner, Hodgkin, Gilbert, Rutter, and Goodman, and Roth, Quattrocassis, Ulrich, and Kahn, who have taken that epochal discovery in Toronto and made it one of the leading areas for future science in the biomedical science area. So thank you so much for inviting me. I'm sorry I couldn't join you in person, but it's been a great pleasure to participate in this conference. Thank you.